USDA Egg Grading Manual, Agricultural Handbook, number 75, Chapter 3, Structure, Composition, and Formation of the Egg. Physical structure. An average chicken egg weighs about 57 grams or 2 ounces. The parts of an egg are the yolk, the white, the shell membranes, and the shell. Yolk, ovum. The yolk consists of the lateral germinal disc, concentric rings of yolk material, and vitelline membrane, a colorless membrane that surrounds and contains the yolk. The yolk constitutes approximately 31% of total weight of the egg. White, albumin. The white consists of 4 distinct layers which together constitute about 58% of the weight of the egg. The caliciferous layer immediately surrounds the yolk and is continuous with the calice, pronounced calice. This is a very thin but firm layer of white and makes up 3% of the total white. The inner thin layer surrounds the caliciferous layer and comprises about 17% of the white. The firm or thick layer of white provides an envelope or jacket that holds the inner thin white and the yolk. It adheres to the shell membrane at each end of the egg. Approximately 57% of the white is firm white. The outer thin layer lies just inside the shell membranes, except where the thick white is attached to the shell, and accounts for about 23% of the total white. Shell membranes. The shell membranes are tough and fibrous and are composed chiefly of a protein similar to that in hair and feathers. The inner membrane is thinner than the outer, and together they are only about 24 ten-thousandths of an inch thick. Shell. The shell is composed of 3 layers and constitutes approximately 11% of the egg. The mammillary or inner layer covers the outer shell membrane. Next is the spongy layer, then the cuticle. Pores connect the surface and the mamilla. The egg as laid normally has no air cell. The air cell forms as the egg cools, usually in the large end of the egg, and develops between the shell membranes. The air cell is formed as a result of the different rates of constant contraction between the shell and its contents. Composition. The egg is an excellent source of high-quality protein and of certain vitamins and minerals. The chemical composition of the egg, including the shell, is summarized in Table 1. White, albumin. The egg's protein content is complete. It contains all the essential amino acids in well-balanced proportions. Water is the major component of albumin. The pH of albumin from a newly laid egg is between 7 and 8.5, and due to the loss of carbon dioxide will generally rise during storage. The thick albumin is made up mainly of proteins: ovomucin, ovobumin, conobumin, ovoglobulin, and ovomucoid. Ovomucin gives structure to the thick albumin. The thin albumin is composed mostly of the same proteins contained in the thick albumin, with the exception of ovomucin. The albumin also contains water-soluble B vitamins such as riboflavin. Riboflavin gives a greenish tint to the albumin. Yolk. The major components of yolk are proteins and lipids, fats. The important yolk proteins are ovovitelin, about 3/4 of the yolk protein, and ovolivectin. The fatty substances of the yolk are mostly triglycerides, true fat 65.5%, phospholipid 28.3%, and cholesterol 5.2%. The pH of the yolk in a freshly laid egg is generally about 6.0, but gradually increases to about 7.0 during storage. Yolk pigments come from green plants and yellow corn that birds eat. The yolk contains most of the known vitamins with the exception of vitamin C. The vitelline membrane is composed mostly of proteins similar to that found in the shell membranes and is fairly permeable to water. The higher concentration of solids in the yolk causes the yolk to increase in size and become less vis- viscous because of the inflow of water from the white as the egg ages. The yolk contains iron, phosphorus, sulfur, copper, potassium, sodium, magnesium, calcium, chlorine, and manganese, all of which are essential elements. Formation. The reproductive system of the hen is divided into two main parts, ovary and oviduct. Most female animals possess both a right and a left functioning ovary, but in the hen, the right ovary and oviduct normally remain dormant, and the left ovary and oviduct develop the egg. Ovary. This is a cluster of developing yolks, each separate from the others, attached to the middle part of the back about midway between the neck and the tail. This organ is fully formed, although very small. When the chick is hatched, it contains approximately 3,600 to 4,000 minute ova, future yolks, each within its own sac or follicle. Oviduct. This is a long tube-like organ lying along the backbone and attached to it loosely between the ovary and the tail. It is approximately 25 to 27 inches long and may be divided into 5 areas which perform certain functions in completing the formation of the egg. It is here that the white shell membranes and shell are secreted. Formation of the yolk. Each yolk within the ovary starts as a single cell, female reproductive cell or germ, with a vitelline membrane around it. The yolk develops slowly at first, but by the gradual addition of yolk fluid. Fat-soluble pigments, normally xanthophyll, are transferred rapidly from the digestive tract to the bloodstream and then to the yolk. Occasionally, what appears to be concentric layers of alternate dark and light-colored yolk fluid may be observed. However, modern feeding and production practices have reduced the frequency of this occurrence. The yolk matures as more yolk fluid is added. The germ stays at the surface of the yolk, leaving a tube-like structure, the latebra, extending to the center of the yolk. An accelerated stage occurs 8 to 10 days before ovulation. This stage is initiated by the stimulation hormone of the pituitary gland. The hormones secreted by the ovary in turn stimulate the oviduct to activity. The rate of yolk secretion increases rapidly from the ninth to the second day before ovulation and decreases thereafter. Ovulation is a release of a mature yolk, ovum, from the ovary. Each developing yolk is enclosed in a yolk sac in which many blood vessels are profusely distributed. These blood vessels carry yolk-building substances to the developing yolk. One particular area of the yolk sac is free of blood vessels, and it is normally here that the ovulation or release of the mature yolk takes place. However, the rupture of the yolk sac and release of the yolk sometimes occur at sites other than the stigma, with the result that one or more blood vessels are ruptured and blood spots occur on the yolk, or the white becomes bloody. Occasionally, reddish-brown, brown, tan, or white spots, commonly known as meat spots, may be found in the egg. It has been demonstrated that meat spots may be either blood spots, which have changed in color due to chemical action, or tissue sloughed off from the reproductive organs of the hen. Ovulation occurs again about 30 minutes after an egg is laid, during the laying clutch of a good producing hen. Formation of the white. The first part of the oviduct, usually 3 to 4 inches long, is the infundibulum, commonly called the funnel. It opens into the body cavity. When ovulation occurs, the funnel engulfs the yolk and starts it on its way down the oviduct. Although the main function of the funnel is to pick up the yolk, it also serves as a reservoir for male sperm, which, if present, fertilize the germ and set up embryonic growth. The yolk moves through the infundibulum or funnel to the magnum in about 15 minutes. Movement is brought about by peristalsis peristaltic action. The magnum area is about 15 inches long, and the time required for the yolk to travel through its length is about 3 hours. It is here that 3 of the 4 layers of the white are formed, and where practically all the protein in the white is secreted. The white contains ovimucin secreted by the magnum as fibers or strands, which makes the whites thick. These strands draw together as the developing egg continues its spiral movement through the oviduct. From these strands, the calciferous layer and the calzate are formed, first layer. The continued twisting and drawing together of these strands tend to squeeze out thin white to form the inner thin white second layer. The remaining thick white is the homogeneous gel third layer. The quality of the white is largely dependent on the amount of ovimucin secreted by this part of the oviduct. The portion of the gel immediately adjacent to the yolk undergoes partial liquefaction before the egg leaves the magnum. The isthmus is a constricted area of the oviduct about 4 inches long, through which the developing egg passes in about 1¼ hours. Here some water and mineral salts are added and 2 shell membranes are deposited. The uterus, a heavy-walled part of the oviduct, approximately 4 inches in length, is where the developing egg spends about 21 hours. Here the outer thin shell, fourth layer, and minerals pass through the shell membranes by osmotic pressure, and the shell and shell pigment are added. The water and solids of the white ultimately become equally distributed in the various layers of the white. Formation of the shell membranes. The shell membranes are added as the partly formed egg enters the isthmus. The membranes are closely knit, lace-like nitrogenous compound similar to that present in chickens' toenails. Formation of the shell. Calcium carbonate comprises about 94% of the dry shell. A hen may use as much as 47% of her skeletal calcium for egg shell formation. 2 layers of the shell are formed in the uterus. The spongy layer consists of small calcite crystals that are not arranged in any order, except in the outer proportion of the layer where the crystals are set at right angles to the shell surface. Pigment, if any, is laid down in the spongy layer of the shell and is derived from the blood. Pores are formed through the spongy layer, connecting some of the space between the knob-like mamilla with the surface. Moving finally into the vagina, a 2-inch area, the fully formed egg enters the cloaca and the vent and is laid. When the egg is laid, the pores are filled by the matrix material and covered by the cuticle. Cuticle, which is sometimes erroneously referred to as bloom, is a chemical composition similar to the shell membrane. The time from ovulation to laying is usually slightly more than 24 hours. About a half an hour after a hen has laid an egg, she releases another yolk, ovulation, and it will likewise travel the length of the oviduct. Abnormalities. Double-yolked eggs result when 2 yolks are released about the same time, or when 1 yolk is lost into the body cavity for a day and is picked up by the funnel when the next day's yolk is released. Yolkless eggs are usually formed around a bit of tissue that is sloughed off the ovary or oviduct. This tissue stimulates the secreting glands of the oviduct, and a yolkless egg results. An egg within an egg is due to the reversal of direction of the egg by the wall of the oviduct. One day's egg is added to the next day's egg, and the shell is formed around both. Blood spots are caused by rupture of 1 or more small blood vessels in the yolk follicle at the time of ovulation. Since the album of freshly laid eggs is usually cloudy, the detection of small blood spots during candling is difficult. Meat spots have been demonstrated to be either blood spots which have changed in color due to chemical action or tissue sloughed off from the reproductive organs of the hen. Although most meat spots are from sources other than blood spots, soft-shelled eggs generally occur when an egg is prematurely laid and insufficient time in the uterus prevents the deposit of the shell. Thin-shell eggs may be caused by dietary deficiencies, heredity, or disease. Glassy and chalky-shelled eggs are caused by malfunctions of the uterus of the laying bird. Glassy eggs are less porous and will not hatch. but may retain their quality. Off-colored yolks are due to substances in feed that cause off-color. Off-flavored eggs may be due to certain feed flavors or improper storage practices.